Hi everyone. I'm working on your recordings for the phylogenetic practice, but I just wanted to quickly kind of cover some of the things from the discussion board and the assignments from this last 2 weeks. Talk about where we're at and talk about your midterm evaluations, some responses to that as well. So let's see. Let's start with troubleshooting for from your midterm evaluations. We saw a few things. I am working on higher quality video. So Eric noticed that too. He's the person who sets up this whole sound studio. It seems like when I record my computer, there's a limitation in resolution. It can't, the recording and my computer do not agree on what is high resolution. So I'm going to start recording not in the studio but straight from my computer. So we just set that all up for an hour, and that'll be coming soon. So that should fix that issue. When you submit answers for your quizzes, I know that some of them, like this last week, the answer was toothless beak on what's the difference between Archaeopteryx and like chickens or ostriches or whatever. I don't know why Blackboard can't, you give it a bunch of choices of answers that are appropriate, and it just didn't know. Some of you randomly got it, just like mitochondrial DNA is inherited maternally. I don't know why it counts some people as wrong, but I go through and look at it. So don't worry a whole lot about that. I know that's annoying. I don't quite know how to fix it yet. I'm working on it. So I understand that that's a problem, but for other quiz questions, especially essay questions, if you missed it, I leave feedback. So if you go back to your grade book, where it says My Grades on Blackboard, and you click the little icon next to that quiz, it should show you the feedback that you get. For your muscle assignment, if you're missing anything, I left feedback. And there's a rubric for assignments, so it shows you which things weren't clicked. So go back and check if you're wondering why you missed something, and see if you can see that feedback. If you can't, there's a button somewhere in Blackboard Cloud that I haven't clicked. But as much as I know, you should be able to see feedback on your midterm evaluation, on your quiz essay questions, on your assignments if you missed something. I leave feedback because it's my intention that you can just redo it. I know that grading-wise, I might not get to the grading those parts very quickly, but I'll get there before the end of the semester. Okay. On the recordings, so right now I'm only recording one screen, but when I do record the PowerPoint at the same time, you can click on that screen and just take me out of the picture. Also, in the future, if I'm doing a PowerPoint and me, I'll just stick to the PowerPoint. That's fine. I personally like to be able to see the person talking, just like how I like you guys to have Abby's, so that I can tell who's who. That's just me. But I will just stick to making the PowerPoint so you can read everything, which is why our lecture is still stuck on my computer, because I'm trying to get it so you can read the whole thing. Okay. Your interview assignment will be great. You've posted them now. And then you're going to be put in these groups. Hopefully you can already see that. So if you go to group, I think you should be able to see 4 or 5 other people's blogs about their scientists. You're going to synthesize all of those. There'll be a separate video explaining that, and I'll send a link out. I got to kind of peruse through them today, and then we'll send that out. And as far as your final project goes, I will start posting things about that soon too. All I really need you to have as of right now is kind of a general topic, so that we can start exploring what do you want to do. You can almost turn in everything as long as it fits the rubric, and I'll show you the rubric. So it's your time to be creative with this, or if you don't want to be creative with it, that's also fine. Also, if you just want to answer something in R, oops, sorry, hit the microphone. If you just want to ask a question and then answer it with R, then your final project's done. That's all you have to do. You have to do the whole R thing and show it to me, and then that's it. So you have a lot of options. Okay, so those are kind of the things from your evaluations that I took. I haven't read all of them because those were just the ones, it was due yesterday, and so those were just the ones that had already been turned in. Let's go to your muscle and quiz 3 from a week ago, 2 weeks ago. On your muscle assignment, when you looked up a gene and you took sequences from it, you told me what the sequences were, like what gene it was. So say it was cytochrome oxidase 1 or cytochrome oxidase B. Those are both mitochondrial genes. Say it was BRCA4, 5, or 6. Those are nuclear genes. But if the alignment showed A's, T's, C's, and G's, that's DNA. If it showed R, K, L, a bunch of other letters, that's amino acids. So that's protein sequence. So I really want you to know the difference between if you're inputting protein sequence and if you're inputting DNA. That's also going to be important for your R assignment. So keep that in mind. Go back and look at that. That is a key, detail that I want you to know. I know that wasn't, I didn't specifically say that, but we're trying to get you keyed into some of this logic, right? So if it's A's, T's, C's, and G's, it's DNA. If it is other letters, it is protein sequence. Now, sometimes it'll be INS, as in the, nano, in. That just means that they didn't know what the answer was. So that's in variable sites. Okay. Yes. Mitochondria came from endosymbiosis, but that was 1 or maybe 15 endosymbiotic events billion years ago, 20 million years ago, something like that. Now, your mitochondria is maternally inherited. There are just some things that are maternally inherited and some things that are paternally inherited, like the Y chromosome. Your Y chromosome looks identical to your father's Y chromosome, if you have one. Mitochondria come from your mother. They're largely conserved or the same because the mitochondria is very important. And so you don't want there to be a lot of variation. And so therefore, it looks almost identical. People use this. I've used this a ton for my, when you study a non-model system, because a katydid has a mitochondria. We know that. We don't know a lot about their genome, but we know that there's a mitochondria in there, right? Or a sea slug, or a copepod, microscopic, or tardigrade. Any of these things, they have mitochondria, so we'll use that, and we can cover large spans of diversity that way. Anything with a mitochondria is going to have cytochrome oxidase 1, probably, because there's 34 genes in a mitochondria, regardless of who you are or what type of organism you are, as long as you are a eukaryote. Okay? And then finally, we didn't touch on Hardy-Weinberg, mostly because you've already done that. Also because we're not going to do a lot of population genetics, because you do that in ecology, you do that in population genetics, you do that in biostats. I love population genetics, but I think we just have other things to cover. So the deal with Hardy-Weinberg is, first it was 2 people, Hardy and Weinberg. This was before the internet existed, so you could come up with an idea somebody already had too, but you didn't know. They lived on different islands or countries, Germany and England, I believe. So Hardy-Weinberg is this null hypothesis, basically saying if everything is random, we will have this many dominant alleles and recessive alleles. It works with one trait that is a single inheritance, and we'll have 2 alleles, okay? representing it. So these are things like widow's peak, or if you can roll your tongue. Those are traits that would be really simply inherited, and you could figure out the Hardy-Weinberg theorem. So it's just math. It's algebra that lets you look at probability. And then you can tell if my numbers are different than if it was random, then there's something afoot, if you will. Something has changed about the system. Maybe there's selection. Maybe the population isn't big enough. Maybe everybody in the population isn't mating and you didn't know that. Something like that. So that's all Hardy-Weinberg is. If you need to know more about that, let's talk about it. Just email me or something. But sometimes we'll make reference to that. It's the null model. It shows random everything. Shows that evolution is neutral and random. Okay? All right. Let's move on to the discussion board. You guys, it was interesting. I've been talking about this I've been talking about this for a long time, this idea of the Human Genome Project. So the Human Genome Project started in about 2002-ish, and it hit some major roadblocks. First of all, sequencing technology has changed by like 600-fold. Sorry, my glasses have a glare. So the technology is completely different. It's way cheaper by orders of magnitude. It's faster, all of that. Okay. Some things that happen. There was a company that used to be called Myriad Genetics. It was like a Monsanto or a Pfizer or some sort of company like that, okay? They were a big part of the Human Genome Project. The original goal was like to sequence 1,000 humans, the whole genome. That took a really long time to figure out how to sequence an entire genome. Two things happen. One, the Myriad Genetics company, who was helping the Human Genome Project, which was through the NIH and the CDC, they patented the BRCA6 gene. So this is one of the genes associated with breast and ovarian cancer, testicular cancer, these type of things. It's a proofreading gene, which means if you get an error in your proofreading gene, then you're not proofreading correctly, right? So then you're building up more and more mutations. So it is early onset cancer is often associated with these BRCA genes. So they patented it. Why? Because nobody said they couldn't patent it. Nobody knew, I don't know, because they thought, well, probably should do this in case this earns us money in some way. Was it completely mean or ill-spirited? No, I don't think so. I think that they just didn't know what the rules were, and so then you get in on this, right? They patented the gene. Then somebody, and it's kind of hidden now, I heard this story from somebody who worked at the CDC, patented the whole genome of a South American tribe. I think because they thought this genome hasn't been exposed to X, Y, and Z, so maybe we can figure out the differences in a population that's never had smallpox and a population that has, or vice versa. But here's the thing, you can't patent people. That's not okay. You can't patent a culture. You can't patent a group, a population of people. You can't patent a population of squirrels. So the Human Genome Project basically hit a huge snag at this point because the rules weren't there to keep them from doing it, so they did it. But it wasn't in good spirits. That's when that GINA, G-I-N-A accord kind of happened. So you can't patent people, you can't patent a gene, you can't patent a whole genome. Also, you are not supposed to be able to use somebody's genome against them, okay? So an insurance company can't say, oh, you're genetically predisposed you have the predisposition for whatever. Now, does that actually happen? I think so still. I mean, you hear stories like Marfan syndrome is this, usually happens to men, not necessarily, but elonged faces, elonged hands, really tall people. Their aorta pump, or their heart pumps so hard because they are so tall that eventually the aorta blows out. So we could screen those people. They could have surgery. They could, to protect that exact spot that it blows out. But being screened means that you are indicated to have it. And I've heard, and I think they fixed this, but I don't know, but we always heard stories about if you did get screened positive for that, then your insurance would go up really high because you have to have all these surgeries. So that's a thing. That's a wishy-washy territory. Let me tell you, we talk about this all the time in my household. So my daughter's father is a scientist for the Department of Energy. This basically means he's a federal scientist. So he works for the country. He works on biofuels, but he's really a bioinformaticist, so he does computational stuff. So he also consults on biosecurity. One of the major players in biosecurity right now is how secure are our genomes. So the ideal thing is you have a card or a chip or whatever that has your genome on it, right? You give it to the pharmacist, he plugs it in, he looks at if you're going to have a predisposition to like negative interactions with certain medications. So they could make a medication that's more specific or tailored to you. That's like ideal. That's easy. That's not far off. It's ideal. The negatives are we're not quite sure how secure is. So not just this insurance company idea, but just in general, what keeps people from inputting fake sequence into a database, a genome database? How do you know that sequence isn't real? Also, then somebody inputs a fake sequence and then pulls it down, like a grad student needs to make a primer, it pulls it down, and then you've created something that could be really dangerous. This is mostly a concern for like bacteria and viruses, smaller things, but it's a weird and interesting idea. So there's people that work on this. There's people that think about this a lot. And that's a major like security risk. Now let me tell you one thing though. When you're talking about the concern of planting DNA to like frame somebody, synthetic DNA is actually one very different than regular DNA. So if you were taking DNA from a blood sample, the people who are doing that, it's not like CSI. Yep. CSI where it's like super fast and then it's colorful and then it all pops up here. It's slow. Takes forever. But the people who are doing that also know enough to know like this seems different, or this blood sample is younger or older or not randomly placed or something like that. So I'm not as concerned about that. Like a lot of people are like, what if I get cloned or they sequence my DNA and then they plant it somewhere? That's artificial DNA looks different. I wouldn't worry about that as much as just large other things. Okay, back to the human genome. So they were getting people to pay like $2,000 or something to sequence themselves, to figure out their ancestry, to figure out what they're predisposed to whatever, right? Here's the thing. The database is now full of just like privileged white people, largely. People who could afford to pay for this luxury of knowing where they come from. That isn't most people, right? So it's a lot cheaper now, I get it, and people really are into it, and so maybe it's getting more diverse, But largely, the Human Genome Project that we know of right now really represents a very small group. In fact, I saw this on Twitter and I should have saved it and I didn't save it. But I read something the other day, like you have a 60% chance of, on one of those ancestry programs, of sending in your DNA and matching with like a first or second cousin if you are a Caucasian person. So the database currently is just all white people mostly. So it's not actually that helpful because we need to know the scope of variation, right? That's true for anything. That's true for E. coli. That's true for, what else have we done? Sea slugs. That's true for tigers. We need to know where there's variation and where there's really conserved traits, okay? That diversity is really important, actually. So the Human Genome Project has some issues, has, had historically some issues, and now we have this new thing with the databases being like linked to your Facebook and all of that, which didn't used to be true. The people who, so the story with the Golden State Killer is that the detective's like wife or something was doing one of these ancestry things, and then they talked about it, and then he was like, hey, we could try this. And so the idea is that like you don't have to sequence yourself, but if we find DNA, you can match it to like some of these programs only match to like third cousins, some of these claim they match differently. It depends on the scope of what's already in the database, right? And you just don't know. We talked about fake DNA. We talked about that. Okay, so when we're on this topic of diversity, we need to talk about, and Ryan brought this up in his discussion board, like it's 2018, so we have to talk about real life things. So we used to just politely skip over the fact that some of these evolutionary biologists, even some of the ones that you guys might have studied for your assignment, were into eugenics. Were some of them into eugenics as a concept, but not as like an actual thing? Maybe, sure. But we don't need to politely ignore this or give it the benefit of the doubt. Eugenics is racist. That's all it is. All it is racism. So we just call it what it is and then move on. Watson, sexist, and it's just all over the place, but also racist. And because he thought at one point some of these, like screening people for a genetic disposition for crime activity or whatever, there's so many aspects of that you don't know about. Also, if we only had this really limited group and we're going to say, oh, these are the fittest people, then that's already like flawed. What we know is, in evolution, the more variation, the more likely a population is to survive, right? Diversity is strength. That's all there is to it. So we've covered that. Eugenics is racist. It's 2018, so we have to say it right now. But that's all there is to it. The greater the diversity, the more likely we are to make it, honestly. We don't know what the next thing coming is, right? That's all there is to it. Okay. Let's see. Oh, Alex was talking about how this idea of whole genome sequencing and using it in medicine or using it in criminal justice system means that we need more scientists. Yes, this is it. Let's get more scientists out in the regular world, right? Not just on this campus. I'm with it. I'm for it. Everything about it. Oh, Sydney brought up this really interesting, and I don't know if you were citing this article, but so there's false positives, right? Those of you who run PCR, you probably run something, you're looking for these, you're trying to amplify these genes in these samples, right? But you probably have one that you know will work, and you probably have one that you know won't work, right? So sometimes people forget to have controls. in England, there was this, like, they were screening, I think they were paternity kits. I don't think they were, like, rape kits or anything like that. But they were sequencing all of these individuals or kit samples, and they kept going to the same person, and they thought they were identifying a serial killer. They were actually identifying somebody that worked at the Q-tip swab factory. And so here's the thing, you should have a negative control. So they should have cut off a Q-tip, stuck it in the, like, DNA extraction kit, and, you know, like they do all the other ones, but this one didn't have a sample on it. That should come back negative, right? But it didn't. And so they sequenced, I don't know how they figured out that it was this person, but that is like basic biology. Anybody that has taken genetics, so all of you should already know that. And there was, you know, it took England a few months to get to that part. Okay. While we're on that aspect of rape kits and various different, like, criminal system sampling, it's getting faster. The more scientists we have that can analyze the data, the better, right? And so there are people now that are putting bodies behind this idea that we can't just store all of these. And also DNA degrades, like it gets shorter and shorter, your sequences, so you have to amplify them more. So when in Detroit, they did, wrote this down, 11,000 kits of samples. They identified, so from 11,000 samples, they identified 817 individuals. So those individuals that they identified had been responsible for all 11,000 crimes, okay? And then there were about 50 of those 800 that had over 10 crimes associated with them. So this type of analysis can be really powerful. You need controls, you need all of those things. But because we can find a new way of identifying patterns, and that is what evolution is about. And that's what all of these, like, phylogenetic practices that you've been doing, it's just ways of identifying patterns. I know it doesn't feel like that yet. I think the next 2 lessons, and then when you work on your final project, you'll feel that a little bit more. But the idea that there were 50 people responsible for 10 to 30 crimes. And then I read a different paper that said they pooled all the data from these kits from, those ones are from Detroit, but Ohio, and Georgia. And they identified individuals that were repeat offenders in, like, they think in multiple states. So there's power in using DNA. Yeah, there are some concerns, but I honestly don't think there are as many drawbacks as there are benefits, because we will get to this more tailored medicine. We will get to this more specific identification, and the technology can only get better, right? But we'll see. Okay, I think that's everything. Megan, I don't know if everybody saw this, but she used the term bad goose lurking. I don't know if that's like a bad goose lurking would be like the things we don't quite know yet about the problems associated with whole genome sequencing or having these large databases of sequences from everybody. So that was funny. I liked it. And in general, I think that you guys are doing really well. I like where we're going with all of this. We're going to take this conversation, especially this genetic predisposition, a little bit further in week 6 or 7 where we talk about epigenetics, because it's like genetic predisposition means you might have those genes associated with it, but then we can start talking about how there used to be this nature, or there still is, this nature versus nurture concept, right? On what you're genetically, or how you behave and how healthy you are and all of these different things. But now we're learning that a lot of that has to do with epigenetics, which is your nature. It's what you're exposed to. So I'm going to kind of push this genetic predisposition idea, keep it, and we'll talk about it more when we talk about epigenetics later. Okay, I think that's it. Good. Keep going. You're doing great. And I will see you soon.